The next experiment that you will be conducting involves the streak plate method. You will be transferring your slant stalk culture, which is this right here, to a plate. To start, you should label your solid nutrient agar, which is this right here, with your initials. Plus, we are also going to be doing a pattern on the bottom of the plate, which I've already done with my China marker, which you can see right here. And once you've created that pattern on the bottom of your plate, you can actually start your experiment. The first thing that you need to do is obtain your culture, but before you can do that, you need to sterilize your oculating loop, because this is what you're going to use to obtain your culture. So we're going to put this in the Bacto incinerator, sorry if you can't see that, but in the Bacto incinerator for about 5 to 10 seconds. Once you've done that, let the inoculating loop cool off, but for time's sake, we're just going to skip ahead. And now what you're going to do is you're going to pick up your slant culture that contains your unknown bacteria, take the cap off, flame the tube for 5 seconds, take our sterilized inoculating loop, scrape out a small amount of the culture, flame the test tube again for another 5 seconds, put the cap back on, and then put the test tube back on the test tube rack. Now that we've obtained our culture on our inoculating loop, we are going to spread the culture across the surface of our agar plate, like so. And in between each spread, you have to sterilize your loop. And the reason behind this is because we want to create a pure culture. We want to isolate these colonies of this unknown bacteria. So that's why I'm doing this. And as you can see, I'm just following along with the patterns that I created at the bottom of the plate. You can also see that when I spread it, I only go from left to right in one direction. And when I'm spreading it, I touch the last place that I spread, just like that. Sterilize my loop again, put it back on the bench. Once you're done streaking and spreading your culture over your agar plate, you're actually going to incubate this plate for 48 hours at 37 degrees Celsius. And when you put this in the incubator, what you're going to do next is move the materials out of your way and disinfect your workspace again. You're going to use germicide to disinfect it, and then you're going to use paper towels to wipe up and dry off your workspace. Make sure to throw your paper towels away in the biohazardous waste container. When you're done disinfecting your workspace, make sure to remove your gloves and put them in the biohazardous waste container as well. Then go wash your hands.